Okay, so we're very pleased to have Dr. Robert Moulder giving the talk titled Target Serum Proteomics of Longitudinal Samples from Newly Diagnosed Youth with Type 1 Diabetes Affirms Markers of Disease. Just a little about him. He's a senior scientist at Turku Bioscience in Finland. This is a research center jointly supported by the University of Turku and the Abu Akademi University in Turku, Finland. He primarily focuses on mass spectrometry-based proteomics research related to type 1 diabetes and the characterization of T helper cell subtypes. And just additionally, a side note, he's from the UK. He graduated from University of Leeds. He had a postdoctoral position in employment in Sweden and now lives in Finland. We will have entertain questions during the talk. You can drop them in the chat, and at the end, we'll have an open discussion. And I've also dropped the two papers that I think you might be touching on in the chat. So you've got them there for your reference. Thanks so much for joining us, Robert. It's really great to have you, and kind of as very exciting times as yourself and your larger group together with Anodia just validated these findings. So can't wait to hear more.
Okay, you're right. Thank you for inviting me here today. And it's been a great time within Anodia, and it's very nice to have these results ready now. So as an outline of the talk, I'll go through the publication, the key publication, some details about the Anodia study and how we got to the work we did present here, how the results turned out, and what future perspectives we hope to find from these results and the samples we've looked into. So here we have again the title of the paper, and then I've marked in all the authors of the paper. And I did try some animations, but for simplicity, I've color coded the authors by institute. So we have myself from Turku, Finland, and then we have researchers in Belgium, Cambridge, and Denmark, all taking part in the Anodia consortium.
It's a great effort with a lot of big name scientists. So it really gives it a big credibility piece.
Also, especially to mention Mikael Kniep in Helsinki, Finland, has been a great collaborator for us here in Turku.
Fantastic.
So the Anodia study, this is a pan-European study. There are 13 countries involved throughout Europe. And the name comes from, if I can speak this out clearly, innovative approaches for understanding and arresting type 1 diabetes. And you can see I've bolded out the and the dia in that to sort of just demonstrate how the name came about. This was funded by the EU and the Innovative Medicines Initiative, plus Breakthrough Type 1 Diabetes and Helmsley. The goal for Anodia was to fight type 1 diabetes, and in fighting type 1 diabetes, there was a key aim to launch clinical trials to slow the progression of type 1 diabetes. Whilst in that process, there was efforts to study the biochemical characteristics of newly diagnosed patients and to identify mechanisms and markers of risk and progression. And just to add, so I don't forget, this is the work we did in the University of Turku in the groups of Professor Rita Lahismar and Professor Laura Elo. And I am very grateful for my joint first authors, Karoliina Hirvonen and Tommi Välikangas. So just a bit more on Anodia. So here's a map of Europe, and you can see some of the sites are marked on here of the 13 countries involved. The study was started in 2015, and already by 2018, already 1,000 individuals have been recruited. So this included newly diagnosed individuals and unaffected family members or first-degree relatives. The last patient was recruited for intervention in 2024, and I think the last part of the trial was just finished in April. And as I said, there are efforts to launch clinical trials. There's a list of these drugs here. You might wonder why there are 4 different drugs, but they had different age groups they were aiming for, the different treatments. And these studies are due to come to the end, and the data to come out as in the fall of this year. With the visits of the newly diagnosed patients and their relatives to the clinic, there was a standardized protocol set up. And the idea was to take all these different samples. I have about 11 different tubes indicated here for different biochemical analysis. And the so-called Inodia protocol aimed to collect these in a highly standardized and appropriate manner. And we'll see details on how well that turned out later in the presentation. So that's just visit one. So it's 6 weeks from diagnosis. And then the newly diagnosed would return at subsequent visits at 3 months, 9 months, 12 months, and 24 months. And once again, there's a whole range of samples taken. So there is a great body of biochemical samples to follow what happens in early type 1 diabetes. As a milestone in the study, there were with the recruitment of the first 100 newly diagnosed patients, there was an effort to do a biochemical characterization of all those samples and bring together that data. So this was called the first 100, and this was like a milestone study. And you see the reference site at the bottom here. This data is brought together to give an indication of how these analytes look like in the newly diagnosed patients. Amongst those methods, there was RNA-seq measurements made of the first 100, and that was also done in Turku. And when we received the samples, we made an effort to also obtain samples which were from the baseline and at 12 months. And so we had a sort of a longitudinal study of the RNA-seq data, and the reference indicated below there indicates the outcome of that study, which some very interesting data came out of that. Then there's the targeted proteomics part, which is my part in this study. And once again, the first 100 newly diagnosed, and we were able to get all the available samples from the first year after diagnosis, and samples from age-matched unaffected first degree relatives. So targeted proteomics. So I'll say a few words about that. So this is a chromatography and mass spec-based technique, and the mass spectrometer is set up in such a manner that you record specific peptide ions, and they are broken and specific fragments are recorded. And by having a selection of specific ions and specific fragments, the technique is very high sensitivity. And at the same time, we have added to the sample synthetic analogues of the peptides we want to measure. So there's very high certainty that you're measuring what you want to see. Targeted proteomics, when it's performed in this fashion, is much quicker than regular proteomics, because you might be looking for a few hundred analytes, and so that is quicker. And just as a comment, so targeted proteomics is not the same as discovery proteomics. Is something that we'll come to later. In discovery proteomics, you try to measure everything you can. Targeted proteomics, you have this certainty, but you only measure what you want to look at. So when we started this, we had to decide, what are we going to measure? So you have to know what you're looking for. So to assemble a list of proteins we could measure, we took a look at the published literature and the data which we had produced earlier. So mostly in Turku, we have been studying samples from the Finnish Diabetes Prediction and Prevention project. So this is samples from children collected before diagnosis. So, for example, the reference Moulder Diabetes 2015 deals with samples from from DIP. Whereas for the newly diagnosed patient, there were several publications from the United States, particularly the work from Thomas Metz at Pacific Northwest Laboratories in Washington, have done some excellent work on characterization of such samples. So if you look at the group of proteins shown in the slide, this is only a subset. You have classic serum proteins, so complements, apolipoproteins, proteins which change with acute phase response, a lot of insulin-related proteins, and then, of course, a lot of coagulation factors. So this was our starting point, and so I'd set up a method to record about 100 protein targets using about over a few hundred peptides. Having received the samples from Enodia, working together with my colleagues and people at the biobank, we made a very tight batch-wise design to remove any bias and block effects from the study, and including quality controls. And then the samples were randomized. So we tried to avoid biases by having a mismatch of male and female samples in a single block, or separating individuals from their own samples in time. Once we'd analysed the data, we did sort of make some decisions of how we was going to work with the data. And as I started out, I said this was the first 100 newly diagnosed, but we decided that we would limit the group to those individuals or the youth who were diagnosed by the age of 18. So it's a sort of newly diagnosed youth. So that reduced the number of individuals down to 86. There was a good balance of male and females, 45 to 41. And for comparison, we had unaffected family members, and that was sort of at a rate of about 2 to 1. So it's 103 males and 95 female unaffected family members. The figure on the right-hand side shows a sort of a schematic of the samples received from 6 weeks through to 12 months, and the distribution of the age and sex of the participants. The comparisons which are made are sort of visualized also at the lower part of the screen, where you see we made a comparison between the newly diagnosed and the unaffected family members, and then a comparison with C-peptide. To achieve this data analysis, a PhD student, now graduated PhD, Tommy Valikangas, used linear mixed effects models for normalization and to adjust for any batch effects. And then these were further used to accommodate for difference from the sex and height, body mass index, and the center of sample collection. What we found was a very useful parameter amongst the clinical data, of course, was the C-peptide data. So now we have longitudinal serum measurements for these individuals, and we also know how the C-peptide changes in time. And as I assume, although I don't know for sure, that all the audience is working with type 1 diabetes, but although if you're not familiar with C-peptide, this is produced with endogenous insulin. So if you still produce insulin, you will find C-peptide is circulating in the blood. And so the levels of C-peptides will show you if an individual with diabetes is still producing insulin, and at what level. And so we have a measure of how serum proteins are changing with time, and how they might be associated with the proteins that we've measured. And working on that hypothesis, from the data analysis, it was actually found that there were 12 peptides, which represented 11 proteins, were associated with C-peptide glucose. So it's fasting C-peptide glucose ratio. So some of these were positively associated, free, and there were also inverse associations. In the bolded text, you can see that apolipoprotein L1 had the strongest positive association, whilst for the inverse associations, the protein glutathione peroxidase free, which is GPX3, had the strongest inverse association. The slope that we observed for C-peptide, as you see, there is this parabolic-like hump, and that's a expected or common phenomenon that's seen, that there's what is called the honeymoon trend in diabetes. So newly diagnosed patients, as they begin their insulin therapy, there is a sort of, for a while, it seems that the endogenous insulin secretion picks up for a while and then declines again. So we have this nice trend there, and you can see it's the proteins which we've selected in the plots actually quite well mimic that trend in the positive and the inverse direction. So with that, and second to that, we did also notice that there were some individuals where the C-peptide stayed fairly stable, and there were others which were dropped off quite quickly. So based on that observation and the associations, we asked the question, can any of these proteins be used to predict how C-peptide changes in time? And once again, Tommy Valikangas looked at all the data. He calculated how much each of the proteins changed in time, calculated the slope for each protein, and then compared individual by individual how that protein changed and how their C-peptide levels changed in time. And what he found was that for one of the proteins, glutathione peroxidase 3, in individuals where the levels increased, the C-peptide levels went down quicker, and vice versa. So this would indicate that the GPX3 is a sort of a sign of the adverse effects, and suggests it could be used to predict the rate of loss of C-peptide in the individuals with type 1 diabetes. Then if we carry on and look at the differences between the newly diagnosed and the unaffected family members, we found that there were 18 peptides from 13 proteins which were differed in the comparison. Some were more abundant and some were less abundant. So I have two examples here. Insulin growth factor binding protein 1, which was at which way did it go? Higher levels in the newly, I can't see my moment, which was at higher levels in the newly diagnosed, whereas ApoC1 was in lower levels in the newly diagnosed. So taking this information together, we sort of sketched out a graphical summary of the changes we observed. So, and then grouped them and sort of the key driving elements. So we had the proteins which were associated with insulin, which of course, or predictably had insulin growth factor families, and then there are apolipoproteins are affected by insulin secretion, so they sort of grouped in there. There are a couple of proteins which are reported in the literature to represent how the islet beta cell functions work and how the integrity of them. Proteins related to oxidative stress and coagulants, and they're reflecting this hypercoagulative feature of patients with type 1 diabetes. So that brought us to our, let's say, our first Inodia proteomics publication, and that was actually, so that was the lead up to the work you asked me to present today. So here we have the full title, the graphical abstract, and the reference as you have posted in the chat. So that was the first 100, and of course the Inodia study was still running, so people were visiting the clinics and samples were accumulating. So with the growing study, this enabled us more material to validate the first 100. So the first 100 is shown here, and then came the next 150. One thing to notice, the next 150, first 100 was, I think, quite carefully selected to get this male-female balance, whereas the next 150 was a bit more skewed, and that influence comes from the fact that that type 1 diabetes is more common in males, so there's a bit of a male bias in that sample group. If we look at the methodology used for these, for the first 100, I started out with a traditional instrument, which is known as a nano LC, and to keep things in a manageable manner, I had a method which took about 45 minutes. But because these were an analysis of serum samples without any other treatment, I had to run blanks in between samples, and this actually meant that my throughput was about 16 samples per day. So that's over 500 samples. If you do the maths, would suggest it might be 40 days, but that's in a perfect world, so don't believe that. But to speed things up for the second set, where we had getting closer to 900 samples, I used a different kind of liquid chromatography instrument, an instrument called the EvoSep. And that's a sort of has been a bit of an innovation in the field. It allows you to load samples in a sort of a batch fashion, avoid running any blanks and loss of sample, and reduce sample contamination. And with that instrument, I could run 60 samples a day. So that would translate into about 15 days of mass spectrometry, but it wasn't. But it was good.
That's Robert, can I interject that? That's a Danish company, isn't it? Evosep?
That is, yes. Yeah.
Thank you.
And that was a much more robust instrument, so it's good for the higher throughput. However, it's a bit less sensitive. So mass spectrometry is concentration dependent, or it's sensitive to concentration. And there was so with the nano LC, the flow rate is about 300 nanoliters a minute, whereas with the EvoSep is increased to about 2 microliters a minute. So there's a bit of a loss of sensitivity because of that element. So if we take in the next 150, this is a sort of the proteomics and data analysis workflow. So similarly, we have samples from 6 weeks through to 12 months, and also from the unaffected family members. The samples are digested, targeted mass spectrometry is performed. We have C-peptide fasting glucose data, and then the data was integrated. Once again, mixed linear effect modeling was made, and we could look at the difference between the newly diagnosed and the unaffected family members. Similarly, looking for C-peptide associated protein changes, and the idea was going to validate these protein levels, and these targets we intend to follow up in the Enodia cohort. So this is the comparison of the newly diagnosed and unaffected family members. So on the left-hand block, first 100, this shows, there's a lot of numbers here. This shows the FDR of the proteins we measured and the effect size. I've put effect size for effect size, so you can see that they are in the same direction and quite generally quite similar. Although you can see for these insulin factors, IGF-BP1, IGF-BP3, IGF-1, I've sort of blanked those out because they were not working well. And here again, this is not, I don't believe this is biology, and this was because we couldn't really get a signal with this setup. So the EvoSep being a bit less sensitive, and our mass spectrometer we used in this instance being moderately old. Some of the proteins, we had additional peptides, more than we had in the first instance, and these further demonstrated that the findings were truer than true, in many cases improving the FDRs quite significantly. So here's an example. The upper panel shows the first 100, and this for a protein which is less in the newly diagnosed, and it's repeated in all 3 peptides in both the first 100 and the next 150. So as a summary, that was the first part. So how did it? So 10 out of 13 protein differences were validated, with additional peptides added into some of these. So we were pretty pleased with that. Then we came to the C-peptide associations. Here things weren't so great, but they weren't totally out of line. So we had 3 out of 11 validations. So for example, if we look at apolipoprotein B, in the first study, part of the study, we saw a negative association with C-peptide glucose. In the follow-up, measuring 3 peptides, we repeated this finding, and with the same trend observed for glutathione peroxidase 3, where we had seen a negative association with fasting C-peptide glucose. In the next 150, we followed this up with measurements in 2 peptides, supporting the finding. And the third protein which met our satisfaction was apolipoprotein M. Here again, moving up from one peptide with an association. The first 100 we found the association was found for that peptide and one additional peptide in the follow-up. So that was the first two components looked at in the follow-up. So then we come to the changes in GPX3 and does it predict progression? So that was a very exciting finding in the first study. Unfortunately, this didn't seem to be the case, and I'll give you some reasons why this might be the case. And we have actually have some recent ideas how we can readdress this comparison. So if now I've put up on the slide a representation of the Enodia cohort. So the first 100 and the next 150. As I mentioned earlier, there were more males than female in the second cohort, but you can see that the age distribution is quite similar, and sex was used in the mixed linear effect model, so it should be excluded from the comparison. So maybe
quick question from the chat: do first 100 and next 150 mean two studies with these numbers of patients studied?
So they are separate individuals. They're both from Enodia, but they are separate individuals and separate unaffected family members. There are two studies.
Great, thanks for qualifying. So
the next sort of checkpoint on this was looking how C-peptide did change in the different cohorts. So for the first 100, it was clear very early on from looking at the data, there were individuals which could easily be classified as either fast or slow progressors. And if you look on the left-hand side, the curve in the grey is represents the C-peptide change throughout the study, and there is this nice sort of parabola drawn out, starting about sort of a moderate level and coming down. Then if we look at the next 150, the effect isn't so clear. It's more of a little bump and just a rapid decline. And this seemed to be the case in the study, that there was most of the individuals just declined. And so if there's some intrinsic difference in the cohort, we don't know why, but so the data wasn't best set to exemplify what we was trying to test. So nevertheless, we moved on with the manuscript, and what we found was very reassuring with this work was the novelty and repetition. So as I mentioned earlier, that's 10 out of 13 targets validated between the newly diagnosed and unaffected family members, and so originally in 86 and then in 146 different individuals across time. And this is important when you think how much criticism is been voiced about the reproducibility of science, and here it is shown in this study that you can reproduce things like this. And a very important consideration here is that these samples are actually collected in 25 different labs throughout Europe, and as I mentioned earlier, the original design in the Enodia study was to have this standardized protocol which was very specific how the samples were collected and stored, and we believe that this has really brought us through to this result, because the sample handling is very important, and so this was a standardized protocol, very well executed. So with the study, you've probably seen this is a graphical abstract of how the thing turned out. So we started off with 21 targets, and we have 12 validated at the end, mixed between these different comparisons. But why end here? So could be so much more we could do. So what could we do next with these samples? As I mentioned earlier, we seem to have a very tightly controlled collection. We have all this data, all the samples, we have the C-peptide data. So what could be done next? We could look at these markers and ask something about the treatment studies which were going on in Enodia. because this was a targeted proteomic study, maybe we should have measured something else. As I mentioned at the beginning, we look back at the literature. Some of that literature was almost 20 years old. Well, maybe not that, it's getting that way. And which is, some time. And it has to be borne in mind that in the past few years, proteomics has really gone for an inflection point. And what I mean here, I can illustrate with some of our recent data. So here in Turku, we've done quite a lot of plasma and serum analysis. And in a recent experiment, we analysed about 600 samples using depletion technique, which we've used for many years, but with different, more recent mass spectrometer, and we was able to identify about 1,500 proteins per sample. That's about 3 to 5 times more than some of our earlier work. And so you're making these analyses and you're finding things you've not seen before in that context, and it suggests you could find a whole new range of biomarkers and a lot more biologically important information. Also, using these samples, we was able to make some analysis made using a special newly designed enrichment system, and what is regarded as one of the top end mass spectrometer in the field, the Orbitrap Astral. And with that work, we was able to identify about 4,000 proteins, and both those two instruments, I'll get rid of that. The Sears enrichment and the Orbitrap Astral are instruments we will now have access to in Turku. So if there's anyone out there listening and you would like to fund our study, please give generously, because I think we can do some really good work with this combination. As I said, we use quite an old instrument for targeted proteomics, a very good, robust and sensitive technique, but Thermoscientific now boasts that you can measure 5,000 targets an hour if you're in targeted mode. And if you're in the field, you don't necessarily need a mass spectrometer. There are all sorts of exciting possibilities where you can measure thousands of proteins with a few microliters of serum. So that brings me to our acknowledgement. So this work was conducted at Turku Bioscience Center with a combination of the groups of Professor Rita Leismar and Professor Laura Elo. The Turku proteomics facility is very important in this. They keep the instruments running for us well and support all our demands, not 9 to 5 or even longer. Of course, the Inoda consortium, and not forgetting Mikhail Kniep. There's a whole lot of other funding agencies that support us, and beyond that, we've worked throughout the years for many years with the Finnish type 1 diabetes prediction prevention study, DIP. So it's always good to acknowledge those guys, because they've been a great help and given us a lot of interesting insights into our work. And that's about the end.
Thank you. Thank you very much, Robert, for walking us through these two very important studies, the discovery phase and then the validation phase, and the great work done by the collaboration piece brought together through Inodia is really amazing. A quick question here from Todd Briscoe, University of Florida. Have the first and second cohort been analyzed with a machine learning using something like RF modeling?
No, we haven't used machine learning on those. No, this was, as I said, done by mixed linear effect modeling, and not in that mode. I suppose the panel is moderately small. I don't know how it would work if you sort of use one of these leave one out cross validation type methods, if there are enough markers for that. I can, that some of the, I don't know if some of the bioinformaticians are in the audience, but I can discuss that with them subsequently.
Yeah, it sounds like something that could be an untapped, way of looking at things. So a great idea from Florida. From Mark Fisher Colbury, it sounds like you might have more data from the discovery proteomics, or is it hoped for in the next step, or is it a hoped for next step?
That's a hope for next step. So we would really like to go through and analyze these samples, and we hope to get that get that going. Of course, we're looking for funding for that as well. You know, things cost money.
Of course. I'll just take a moment to reference Carsten Bouchard, who's actually here. And he's been talking about the role of sphingolipids for decades, mostly in 2018 and several other papers, abnormal islet sphingolipid metabolism in type 1 diabetes. And of course, you're talking a little bit about that with your biomarkers here, and the ApoM and ApoB. So I'll let him take over and ask his question.
Yeah, I just the Danish company, you call it Evocept, or what was the name?
Yes, Evocept.
Evocept. Evocept. Yeah. Okay. Yeah, I think you. Where do you have it?
It's in the paper as well.
Where did I put that? So it's on here, Evocept. So
yeah, Evocept. Yeah. Evocept. Yeah, I think you should measure also the sphingolipids or other things than just protein. And we have been active with these sphingolipids, especially sulfatides, since 1990, but no one has really taken it up. It has several interesting properties. It is at the surface of the beta cells, and newly we have shown that it stopped the fibroblast growth, so there's no fibroblast growth in the islets. There's in the femur when you put in insulin, inject insulin, there's fibroblast and so on. And there are several other interesting properties that it is working against T cells and so on. So it might be interesting to measure these as well, but I do not know whether this company that is measuring proteins only also could take that. But I think you should extend your research also to this. You do really have a lot of money. We are more idea-based, but I think a lot of the same thing has been done for years, and no one has really taken this sphingolipids up.
Well, Carsten, if I could just add from your 2018 paper that was the abnormal islet sphingolipid metabolism in type 1 diabetes, you found reduced amounts of sulfatide, 23% of the levels in the control participants in the and in the pancreatic islets with newly diagnosed type 1. You looked at those 8 gene polymorphisms, the ORMDL3 and SPHK2, et cetera. I wondered if those data are available in these cohorts to kind of dig behind the individuals that showed these changes and see whether or not they carried or they had those gene polymorphisms that you cited in your earlier paper, Carsten.
Yeah, that could be interesting to know. It is published, and I think there's also supplementation in this Diabetologia 2018, and that was really a paper we liked and are proud of. I think you should take these things up as well, and not only proteins.
Yeah. Just from Todd Briscoe, we'd published some work on IGFs. I would not give up on that pathway as it's a surrogate biomarker. He's dropped the link to the paper in the chat, and hopefully newer technologies will lead to replication there. Great talk. Yeah, it does seem that these newer technologies and advances in both the the hardcore lab work as well as the ML AI component are expanding the knowledge and the increasing the possibility for replication faster.
Have I stopped sharing now?
No, you're still on there.
Okay,
I just wanted to ask you, what are your thoughts on how you might want to get a window into these sort of biomarkers for those who are stage 1 or 2 or even 0, prior to diagnosis? Can you start looking into those windows, or is that something we should be waiting for to drop in the fall of 2025 when Anodia releases more of data?
Well, we did look at the unaffected family members, and then we did look at some of the antibody positive unaffected family members. We haven't followed that up entirely in proteomics. There was some work of the RNA-seq work on the unaffected family members, and there are those which have been those which are antibody positive and then revert to antibody negative, which was an interesting subgroup. But when that was done in the first 100 for the antibody positive, but we didn't have enough within 100. We didn't have enough when the UFMs. There wasn't enough power for that, and could be repeated, I suppose, or taken up again.
I guess I'll just throw out the $1 million question. How can you scale these findings to the clinic?
I suppose the thing is to hit on the right marker, and then to think about developing quick tests for which you might sample, or just to follow in the patients under treatment. Actually,
those are who are taking teplizumab, do their markers change, etc., or whatever combination therapy they're eventually going to be exposed to.
Yeah, but I should say that it's a remarkable, great study you have done, and of course, all new knowledge add up to our understanding of type 1 diabetes. So, not to be too critical, which I might be before, I think it's a great study, and congratulations with that.
Right, thank you very much.
Yeah, it's fantastic. Anodia is really pushing the needle in Europe, and it's fantastic to see how so many top researchers like yourself are coming together, together with them and really making things happen. So thank you very much. And thanks for the great presentation. It's really interesting, and I'm sure it's going to have a lot of views. All right. With that, thank you again. We've got a lot of accolades in the chat with nice work and clapping. And thanks again, and have a great rest of your day. We'll be in touch again, probably in the fall 2025.
All right. Thank you very much. Thank you. Right,
Bye-bye.
bye.
Yeah.